Well, good morning, everyone. It's always a pleasure to give presentations, and I always say I'm happy to be here, but we are in Bermuda. It was a special meeting today. Thank you, Joe, for the fantastic talk this morning. 3 things that jumped out at me that will kind of link in, I hope, with the presentation I'm going to be giving is this notion of it's really important for us to use the science. Our goal is to use the science to make informed decisions, not to ignore the science. But we have a challenge to make sure we can use it in a way that we have confidence. So I'm going to talk a little bit about some of the evolving science and how I think that's improving the way that we can handle some of these challenges that Joe mentioned. I also wrote a note down here that we can improve our ability to quantify risks, and that's going to be very crucial in our ability to make some of these judgments about risk comparisons. And that's a challenge we have right now. So we need to get to the next level in terms of our ability to address scientific risk assessment science in informing decisions. And hazard-based approaches do have a lot of appeal in terms of their simplicity, ability to communicate, but they have many limitations, and from using the science perspective, we want to go with risk-based approaches when we can, but we have to make sure we mature our science in a way that makes it usable. So I do have a few examples that we'll kind of weed or blend in through the presentation today to hopefully get that across, that there is hope that we will achieve some of these goals. So my overall objectives for the presentation today, I really want to talk about very briefly the elements of chemical safety assessment for food-relevant chemicals, and Joe already did a great job of sort of laying out some of the main principles there. I will get into the weeds a little bit just to show how one particular area of the science is really evolving. Describe ways to improve the science that are really affecting our methods for safety and risk assessments. And then identify what are the impacts of all of these improvements. Why are we going through this activity and effort? And again, I think that'll be maybe one of our panel discussion points. Is this, if we achieve all the things we're trying to achieve, will we actually be successful in getting to where we want to be in terms of informing risk-based decisions? All right, so this is my layperson's version of the safety assessment process. It really aligns really closely with what Joe mentioned, though. So the way I think about it, just in communication, is, look, we all know that as toxicology people and food nutrition scientists, that at some dose, things are not going to be safe. There's going to be some level of harm that can be caused by everything, whether it's pure drinking water, table salt, you name it. There's a region that's not safe. We know that. I think most of us would agree that there's a region that's safe, or at least a region that has very minimal risk. So since we can kind of probably most people will agree just on common sense of those principles. Now the challenge of us as scientists is, okay, but we will need to make decisions somewhere in between there. So our challenge is we certainly don't want people falling off the cliff, but we don't want to be so far back from the cliff that we don't get any view of the vista. So how do we optimize where we need to be? How do we find out that safe dose region? Well, it gets complicated, right? Because we're using our biology knowledge and our risk science knowledge. We know that in toxicology, chemicals cause multiple effects usually. So the question is, well, which effect is going to be the driver for our danger or our risk? So that's what I kind of call the cracks in the cliff here. The problem is I don't have perfect insight as to where those cracks lie. And because there's a fog of uncertainty, our scientific uncertainty around where those are. So we have a lot of challenges ahead of us. So what we have to do is sort of bring the sunshine in and clear out the fog the best we can to actually identify where these risk cutoffs happen. And we will call that the ability to enhance our ability to do risk-based decisions based on safety assessments. So putting that in the sort of the nomenclature of chemical safety assessment, our first equation here, this again is that idea of an allowable daily intake, tolerable daily intake, etc. This idea of a safe quote-unquote dose relies on some measure of dose response. Well, what does that measure? Well, remember that's the cracks in the cliff. Where is the crack? Where is that no observed adverse effect level? But because there's uncertainty around it, we want to take that into account by moving away from the edge of the cliff. We do that by dividing by some number safety or uncertainty factors. And how do we use those? Joe already mentioned was in the hazard quotient concept, or margin of exposure concept is a similar thing, where we're comparing essentially the exposure level and the safe, quote-unquote, safe dose. So that's the overarching principle that's used and embedded in a lot of these types of risk assessments or safety assessments. So sort of a traditional approach would be I take my toxicology studies or epidemiology studies and I sort of lay out as the dose goes up, I start to see more and more effects. So if you notice here, that first oval, you'll see an NOEL. So there'd be a dose that was tested that really didn't cause any biological changes at all. The next dose up, that NOAEL, we actually started to see some biological changes happening, but they were judged to be not adverse to the system. The next higher dose up would be this lowest observed adverse effect level, we call it. So that'd be the first dose where we actually saw adverse effects occurring. And you can see sort of the blue marked oval there, some changes in histopathology in the liver, for example, and some indications of inflammation. And the next highest dose over the frank effect level, or FEL, that'd be where we're starting to see some real serious things happening. So our goal is to weed through, might be hundreds of studies, to sort through all of this to identify where do we start to see effects occurring. And we'll pick our no observed adverse effect level, then we'll divide it by a series of uncertainty factors to calculate our ADI. And that approach, as Joe had mentioned, has served us very well. Now the question is, what it doesn't do is it doesn't tell us about risk above the ADI. It doesn't actually give us risk estimates, but it can help be used to characterize or make risk-based decisions. So we want to continue to improve this overall process. This is just a slide kind of indicating those areas of uncertainty that we build into the process, and whether you're working related to the World Health Organization or EPA or FDA or occupational scenarios, most organizations do always think about at least these 5 things. They think about differences if you're extrapolating from a toxicology study in animals. Are there interspecies differences? Are humans more sensitive? We want to account for that possibility. How variable is the human population? Are we making sure we're covering the sensitive subpopulations in our risk assessment? What if we actually started with a dose that actually caused things to happen? Well, we're not trying to set a dose that causes things to happen. We want a safe dose. So we might need to extrapolate lower based on that and do that with an uncertainty factor. Subchronic to chronic extrapolation, what if I had longer study? Do I have to worry that maybe the safe dose would have been lower? And the last one is kind of sort of an omnibus factor for database. Maybe I didn't even test for the right effects in the first place. There's more sensitive effects we have not even identified. So these 5 areas are embedded in almost every safety or risk assessment for a chemical that you're going to come upon. There's some others and some variations of these, but these core areas are covered. So that's basically kind of what our traditional approach has been. But the good news is, in addition to that traditional approach, which has served us very well, we are continuing to improve that approach. And what I really love and I'm happy to say is, it's not like we're taking the traditional approach and throwing it to the side and saying, oh, now we've got new toys. What we're really doing is using these new methodologies to enhance the traditional approach. So there's a really nice way of kind of blending the two together, and hopefully you'll see that in some of the examples here. And what we're basically doing is improving our crosstalk across the field. So better discussion with nutrition, better discussion with statisticians, a better discussion with chemists. How do we integrate all of these fields together in making better risk and safety assessments? And I think you'll hopefully see some examples of that. So we are really using our understanding of biology and nutrition, statistics and chemistry to improve the way that we're doing the assessments. And as a traditional sort of toxicology risk assessor, I really enjoy those interactions, and I think it gives us, better products in the end. So what I'm going to do is highlight some of these points with just a number of different examples. So hopefully we don't get too far in the weeds here, but I really keep in mind my main point here is to show you some examples where the science is improving. We're able to integrate that science into our process, and I think we get better, more confident results. So one example is 3-MCPD, monochloropropane diol. So it's a contaminant formed primarily through refining of edible oils, and it's been a subject of a lot of risk assessment controversy and issues. A variety of different groups have developed safety assessments or risk assessments for this particular chemical. So one sort of evolving part of the science is all of the most recent assessments have used a benchmark dose modeling approach, and I'll show you what that means here in a minute, to derive estimates for tolerable daily intakes. All of the groups, and this is actually using similar data set among all these different groups. So the question is, how do we do a better job of characterizing what we're seeing in this toxicology study? There's actually a couple different studies, but so these days, actually, in most risk assessments, this benchmark dose modeling approach is actually now considered sort of the standard approach to use. So what the benchmark dose modeling approach says is, what you see here are the diamonds are going to be the actual fraction affected or response at different doses in a toxicology study. So what you can see with the red line is sort of a statistical best fit curve of those data. And then you'll see that what we can do is we can decide for the different studies what response rate, it's called a benchmark response, do we want to kind of use as our starting point. And that's the blue line. So if we start at, say, 10% additional risk, we can move across over to the red line, then move down, and you'll see that BMD. So that's the benchmark dose. So that's the dose associated with, estimated dose associated with a given level of response. And you'll see there's something called a BMDL, so that's a lower bound on that dose. So that's sort of the statistical bound that's lower. So if we were doing a traditional risk assessment, our starting point, that NOEL, would be 100. Using this sort of approach, our starting point is 50, usually for a risk assessment. Now, they're not always lower, but the point is, as opposed to relying only on the study doses, we're actually relying on all the dose response data, the whole curve, to give us estimates of where our starting point for the risk assessment should be. So it's we have much more confidence in this approach and this method. And like I said, that's actually been sort of the standard approach. This is what the actual modeling software looks like if you do it in the most typical software suite. This actually, US EPA has a free downloadable software. So if you like to do dose response modeling, it's called Benchmark Dose Software. You can download this from the EPA website and run these models. So this is an actual model run for the data on renal hyperplasia, so a renal toxicity effect for 3 MCPD. So you can see how it really looks in real. The other one's sort of an idealized. But again, you can see the curve fit, you can see how well it fits the actual data points, and you can see the BMD and BMDL estimates there. So that sort of approach is good because it's using the data better. So this is that idea of let's not ignore the science, let's try to use the science and inform our judgments. So I think we checked that box for this assessment. But even when you do that, there still can be challenges. So you'll notice here we have several different assessments that have been used this benchmark dose modeling approach. And you can see the answers aren't the same. So what I would say here is another kind of step forward is what traditionally was done is sort of, I'm going to run different models and pick one of those models as what I think is sort of the best choice. And the decision rules for that weren't always the same. So what's happening, though, as you can see, the third column over this says Haber 2018. At the bottom, it says model averaging. And the F says brand new assessment, says model averaging. So what we move towards is the field is moving towards taking a combination of models and averaging those results. And what you can see if you look at this data closely is that starts to converge the differences among different groups. So we're achieving better use of the science, better capturing of uncertainty in the models through model averaging, and in the end, that's starting to move things a little bit closer towards harmonizing or at least increasing the agreement of different types of assessments. So there's a lot of variety available for this particular assessment that we have to sort through as scientists, but we are making progress in how we're using the science, and this is, I think, a pretty good example of that. Okay, another example would be looking at the dose response for trans fatty acids and its effects on low-density lipoprotein cholesterol. So again, the idea here is let's take better use of all the science we have and characterize actual dose response behaviors. So in this particular case, the challenge was we had a variety of different studies, and if you look at individual studies, you get one answer. If you look at different subsets, you get a different answer. And because of the nature of the studies, it was really unclear what the shape of the dose response curve was down at the lower range of intakes. So this particular project was looking at what if we use a sort of a Bayesian statistics approach that allows for calculating the bounds of confidence in different model parameters. So what's the range of the models? And using that kind of approach, what type of model shape or curve shape actually fits the data the best? So that was done using data sets related to cholesterol and trans fatty acid intake as a function of amount of total energy intake. And of course, why is that important? Well, that's selected as an important biomarker for coronary heart disease. So isn't this a cool picture to look at? the key finding here is, again, using this sort of dose-response approach, what we actually were able to show, or my colleagues were, is that actually the best description of the data is not a linear curve, it's actually an S-shaped curve, a sort of a hill function curve. So by using a meta-regression technique where we're weighing all of the data and allowing the Bayesian approach to optimize what the curve fit is, we actually found that this sort of curve shape actually fits the data better than a traditional linear type of approach. And that was supported by the underlying biology, the mode of action thinking. So now we're integrating our biology knowledge, our ability to use advanced statistics together to help inform what we think is better science. This has very important implications, of course, because using data like this really looks at the idea of, well, at what point do we really start to worry about trans fatty acid intake and its impacts on cholesterol? So that's a really important question. If you just assumed a linear relationship all the way down to zero, that would be a fairly different thinking than this particular model shape, which fits the data better. So in addition to statistics, from this prior example, especially in the next one I'm going to show, one of the big advancements is we're using our statistics better, but we're using our biology better. And I think this is an important role for toxicology, pharmacology, and nutrition to really come together, especially as we're dealing with chemicals that are naturally occurring in foods, for example. So one way to sort of organize our thinking around the way that a material causes effects is something called an adverse outcome pathway. Now, we used to have a version of this was mode of action analysis. But what we're really doing is trying to map the pathway between chemical exposure, how your body handles that, the pharmacokinetics or toxicokinetics, and then the series of biological steps that are happening, in sequence or as a pattern that lead to some kind of effects in individuals and populations. So the key thing for us is, as biology folks, is how do we start to identify and map out what are the key biological steps that lead from the beginning to the end of this pathway? Now, the great thing is, if we can start to understand that better, which we are, then we can also start making some judgments about, okay, well, what is the underlying dose-response behavior for the steps? And if we can start linking the dose-response behavior for the steps to each other, we can start arriving at better conclusions about the likely dose-response behavior for the whole pattern, from all the way from exposure to disease. So as Joe was mentioning in his talk, how do we start to get better at informing the actual dose-response behavior? Using mode of action biology as embedded in our dose response is, I think, going to be very important and key to doing that. And in fact, we're already starting to see that happen. And this is an example of using the adverse outcome pathways. The other evolvements in this field of adverse outcome pathways is, well, there are many chemicals that cause liver necrosis or fatty liver, et cetera. Can we kind of map out common pathways or common steps in the biology that are sort of agnostic or don't really matter what the actual chemical insult was? So we can start to leverage our knowledge based on just understanding biology of disease and pathobiology as part of this overarching process. So we're pretty excited about using these techniques as part of embedded in our decision process along with the statistics. And an example of that would be acrylamide, where acrylamide is present in heated carbohydrates. So another one of the, I think is that a category B, Joe? Chemical in the Joe Roderick scheme. So another challenging assessment, right? So it's a demonstrated tumorigen in rodents, mice and rats, but the mode of action is really uncertain. So how does, for example, in rats, that's where at least I've looked at that data more closely. What's the underlying mode of action for the tumors? Is it because acrylamide gets metabolized to glucidomide and that directly reacts with DNA, causing things like DNA mutations? Is it reacting with glutathione in the cell and reducing the ability to withstand oxidative stress in the system and causing oxidative stress, which could itself cause DNA damage or growth signaling? Is it some kind of modified hormone metabolism? Increasing the metabolism of thyroid hormones, for example, and changing the balance of that system. Well, this is all understanding this biology and using it's very important. It has a lot of implications. Right now in California, the Proposition 65 law and limits are under a lot of hot debate related to a lot of different products and what's the safe or minimal risk level that ought to be used for something like acrylamide. So this is very apropos to actually making decisions today in the regulatory context. Mode of action concepts can help us improve our dose response and make judgments about how we're going to integrate all of this complex biology to make predictions. So this is just an example for thyroid tumors associated with acrylamide. And what you'll see here is sort of a traditional approach would be maybe to fit a standard line or curve through the tumor data to arrive at our best estimates of risk associated with acrylamide exposure. What you'll notice in this particular, at least the dark line here, is it really has two different regions or slopes. And this particular model was trying to take into account the understanding of biology of acrylamide's effects, with the idea that at some point you start to actually get a higher incidence of tumors secondary to things like at some concentration where you really start to deplete glutathione, you're actually getting interaction of multiple mechanisms that are having more impact. So you're getting a faster tumor dose response at the higher levels than at the lower levels. So again, this is just an example of trying to integrate biology into our dose response with the aim of improving the way that we can characterize our risk and having more confidence in that it's actually capturing biology. And hopefully this works towards fending off the idea that we have to just go with hazard approaches because we just don't have the understanding to do a good job on a risk-based approach. All right, I'm switching gears a little bit here, but this is the idea. So I've talked a lot about how statisticians are helping us and how biologists are helping us. Well, the chemist should not be left out, so we need our chemists. So here's the thing, okay, I have these two molecules. Is one safe and one toxic? And how do you know? So the good news is we're still relying on our fundamental science of statistics and biology and chemistry to help us out here. There are a lot of potential features in a molecule that could help us make a judgment. There could be structural fragments or pieces of the molecule that we say, hey, whenever I see that piece of a molecule, I happen to know that really likes to bind to certain proteins, and therefore I'm going to flag that as a concern. Or it could be things like chemical or physical properties, like is it soluble? Does it get across cell membranes? Is the molecular weight so big that it is not very bioavailable? Are there molecular properties like reactivity with negative or positive charges that are likely to in biological reactivity properties? So we can start to map out literally hundreds of different features of different molecules, and using statistical approaches and expert systems, start to make some judgments about is it likely that this chemical is a concern or not. So although this is not really enhancing our dose response based on the biology, this is doing another important function. It's helping us find data or fill data gaps where we don't have existing data using these types of techniques. So here, even if you go with the idea of hazard-based, well, I think we have to just not use this chemical. This is a tool that lets us say, well, wait a second, maybe we can find some analogs to our chemical that do have good dose response data, and we can use that information to help us make informed risk judgments. So this is a really important step forward. There are a lot of tools that are available to us. Probably the most common, there's a lot of different tools. Some are commercial, some are free. If you're interested in this topic, though, you should at least become familiar with the OECD toolbox, because that's sort of a big resource that's in this field right now. I've been directly involved in the issue. I don't know if anyone here has dealt with butter flavorings, but the handling of butter flavors, not really identified as a concern for many years as a food ingredient. The oral toxicity is actually quite minimal, but workers were found to have important and serious respiratory diseases who happened to be handling butter flavors, and that was primarily attributed to the component diacetyl. Inhalation, that spurred a lot of activity to try to find out, well, what's this quote-unquote safe amount for worker exposures by inhalation route? So inhalation-based limits for workers range from as low as 5 parts per billion in the air to 200 parts per billion from different organizations. But this emphasis on diacetyl caused a desire to identify substitutes with lower toxicity, and one way to do that is to use read-across approaches. So if I have this thing, and I say, well, gosh, I'm finding this to be pretty toxic, I need to switch to something else, but guess what? The things I'm going to switch to also don't have any data. Now what am I going to do? So what do? We can use our tools of read-across and structure-activity relationship to help us out. So here's a question for you guys. This will be part of the exam. Are these equally toxic potency? So the first one, diacetyl, you can see sort of that you have the red highlighted structure. That's sort of a key structural feature that relates to its ability to interact with certain proteins and cause, we think, cause the primary toxicity. You'll notice that the one at the bottom was really viewed as a good substitute for quite a while, pentane-dione, but you'll notice it has that same fundamental structure. It turns out it actually has about the same toxicity and about the same potency even. So it does not really a very good substitute. Whereas there are some other compounds that maybe not quite as efficacious, but they do have lesser toxicity. So we can use these sort of tools to help us in our overarching judgment, even from a hazard perspective. Overall conclusion then, our traditional approaches have served us well for an adaptable approach to assessing food-relevant chemical safety. So we have approaches that we think work pretty well. I think Joe covered those really well. But that doesn't mean we give up on trying to improve. So we have new science and technology has allowed us to improve the ability to refine these assessment methods. And what's the impact? I think we'll talk about that in the panel discussion, but really to me, we don't eliminate uncertainty, but we can reduce and understand and characterize the uncertainties we have. And really a key feature, because sometimes that final answer doesn't actually change very much, but you can certainly have much higher increased confidence in your assessments. And I think that gets to Joe's last points about if we have high confidence in our risk-based assessments, it really helps us make the case for using them as opposed to low confidence hazard-based assessments. So I think that's my last slide. I should just note that the case studies I presented here worked with a team led by Dr. Lynn Haber and some other colleagues of mine, and some of those case studies were developed with funding from GNA and ILSI Food Chemical Safety company members. Thank you very much.